Good afternoon, everyone. First of all, I would like to thank the organizers for giving me the opportunity to present my work here. So the topic of this presentation is designing the plane spacing for multifocal plane microscopy. And this study is motivated by the 3D single molecule tracking problem. So as we all know, 3D single molecule tracking is a very important tool for understanding protein dynamics in live cells, and it has many applications. So here's an example in which we track quantum dot labeled antibody molecules undergoing endocytosis in a live cell. And as you can see, we have different types of behavior. It undergoes diffusion, for example, in here. We have highly directed movement. If we want, we could estimate the 3D diffusion coefficient, for example, for this part. Now, I would like to point out the key requirements for being able to study this protein dynamics in their full detail are three-dimensionality and also covering large depth. So ideally, we would like to have a 3D imaging modality which can cover a very large depth. So this 3D single molecule tracking relies on a 3D localization procedure. To be a little more specific, we usually acquire a sequence of images at different time points, and we segment them such that there is the image of an isolated single molecule in each ROI. Then we fit a 3D PSF model, for example, one on both PSF model, into the data as shown in here to estimate the 3D location of the single molecule, X0, Y0, and Z0. And we could do it for different time points, and we use a trajectory linking algorithm and finally end up with a 3D trajectory like this. If we use conventional wide field microscopy, we usually have problems on estimating the Z position of the particle. Here's the reason. So this is the conventional wide field microscopy. As you can see, it has only a single focal plane. And we usually have three problems when estimating the Z position. The first one is, we can explain using this image, and let's list what these are. So these are the simulated images of a point source at different Z positions along the Z-axis, and here's the focal plane. So the first problem is about axial symmetry. Most of the PSF model are axially symmetric. It means that if we look at, for example, these two parts, negative 600 and positive 600 nanometers, they look identical. So there's no way to figure out if the point source was above or below the plane of focus. Now the second problem, which is more important, is the depth discrimination problem, as shown in this part. As you can see, when the single molecule is close to the plane of focus, for example, at 150 nanometers, its image profile is very similar to the image profile when it's at the plane of focus. So for this region, we cannot estimate the Z location accurately. And the last problem, which is, I guess, the most important one in most biological application, is that using this conventional microscopy, wide field microscopy, we can only cover a limited depth. So we're not going to be able to cover, to understand the protein dynamics in its full detail. So there are different approaches to address this problem. For example, we have astigmatism, we have PSF engineering such as double helix PSF. We have interferometric optics and multifocal plane microscopy. I'm going to focus on the last one, multifocal plane microscopy, or in short, MOM. So this was developed back in 2004 in our lab, and so the way it works is very simple. We made a few modifications to a normal conventional microscopy setup. So what we do, we have a beam splitter in here, and we have another detector which is slightly shifted with respect to the design focal plane of the microscope. And what we end up with a shifted focal plane in the object space. Okay. This is a two-plane MOM setup. So for this, the observed data will look like this. So we have, so this is the Z-axis. We have a pair of images for each Z position of the single molecule. And that pair is unique when you compare it to the other pairs. So using this, we could estimate the Z position more precisely, and means that we can actually overcome the depth discrimination problem of conventional microscopy. Now this is just a two-plane MOM setup, but there is no limitation. We could increase the number of planes to P planes simply by adding more beam splitters and more slightly shifted detectors. So this implies that we can actually cover a large depth if we increase the number of planes. And in that case, we would have, instead of having a pair of images, we would have P images per Z position. We need to process that. Now, the natural question arises as to how to design the plane spacing, because that would change the accuracy with which we can estimate the 3D location. And also, what is the appropriate number of focal planes? For example, it seems like if we increase the number of focal planes, we can enhance the accuracy. And the reason is that because it seems like we are improving the spatial sampling in the z direction. But remember, we also split the photons using these beam splitters. So it gets a little bit complicated, and this is what we're trying to address in this research. So before addressing those two questions, I would like to first show you how we process the data and estimate the z position in our lab. So what we do, we have an algorithm called MOMLOC, non-localization algorithm. So we basically have a maximum likelihood estimation procedure. So we estimate so let's say this theta hat is an unbiased estimator of a non-parameter vector. We find it such that it maximizes the log likelihood of the observed data. And that log likelihood is just a logarithm of the joint distribution of the observed data. And the reason that this is right is because the data acquisition at each pixel is independent of the others, and also the same for the focal planes that are independent. And the positive aspect of this maximum likelihood estimation is that it's consistent and also it's asymptotically efficient, meaning that over a repeated experiment, as the number of experiments goes to infinity, it's unbiased, and also its variance is smaller than any other unbiased estimator. Now this brings us to the notion of localization accuracy, which is explained here. Or location estimation accuracy. So most people use the standard deviation of the estimator over repeated experiments to characterize the accuracy. But this is typically problematic, and the reason is that this depends on the specific estimation algorithm. And also, usually there is no analytical expression for that standard deviation. And there is another tool in information theory, which is well known. That's the primary role bound, or the CRW. It's basically this inequality. It's simple to understand. It's very powerful. So basically, it says that the covariance matrix of an unbiased estimator of your unknown parameter is bounded from below by the inverse of something known as the Fisher information matrix, or FIM. So this is very interesting because it means that assessing the best possible localization accuracy boils down to the calculation of this FIM, the Fisher information. So this FIM has a very general equation, and it's been used in many different applications. But we have derived an equation for that specifically for single molecule microscopy over years. We consider the practical data model, which we have a pixelated detector, different types of noise sources. And we end up with this equation of the practical data model. So that psi that you can see, that's called the noise coefficient. It depends on the detector type, depending if you're using a CCD camera or SCMOS or EMCCD, there is a separate equation for that. And we have that mu, that's a function of photon count, and the spatial distribution of the photons, which is closely related to the PSF. Remember, this Fisher information matrix is a 3x3 matrix right now because we are dealing with a 3D localization problem. So we can actually pick the main diagonal elements of this matrix, which are shown here. So if I pick the main diagonal elements and name them X naught FIM, Y naught FIM, and Z naught FIM, they provide information about the location of the single molecule. So if the quantities are larger, it means that better we can localize the molecule more accurately because we have more information. Correspondingly, we define the practical localization accuracy measure, or the PLAN, defined in here. That's basically the square root of the main diagonal elements of the inverse of the Fisher information. Okay? So these are in nanometers, and they're actually the accuracy, or some people call it uncertainty. So if it's smaller, it's better. We could estimate the location of the single molecule more precisely. So now we have all those definitions. Let's see how we can calculate the plan for a single plane setup. I mean, the results are shown here. So this panel, the right-hand side shows the lateral plan, or S naught or Y naught plan, versus the object's in position, and that dashed line is the location of the plane of focus. Now, as you can see, when the single molecule is at the plane of focus, we get very low numerical values, means that we can accurately estimate the location of the single molecule. As the single molecule moves away, it gets worse, and it's kind of intuitive, so I'm not going to explain it further. But if you look at the axial plan, so this is the axial element, or the Z the object Z position. As you can see, we have a very large value very close to the plane of focus. And this is actually the depth discrimination problem which we're going to talk about. So now we have a method to quantitatively look at it. So this was for a single plane setup, but how do we do it for a MOM setup? It's very simple, considering the fact that the data acquisition at each focal plane is statistically independent of the other planes. So we can simply write down, express our film for a MOM setup as the sum of the films of the individual planes. So using this, we can actually show that this MOM can overcome the depth discrimination problem, and it can cover a large depth, which are shown in panel A right here. So this is the axial plan versus the object Z position again. The dashed lines are again the location of the planes of the focus. We have two focal planes now. And I would like you to focus on the black curve for a second. So this is the plan, the axial plan for the MOM setup. As you can see, it's pretty flat and it has low numerical values. So we don't have those large values anymore. We solved that depth discrimination problem. And the reason this happens can be explained using panel B, which is the axial film versus the object Z position. And so this film is inversely related to the plan. So a small value for the film corresponds to a large value for the plan. Now, if you look at the blue curve, for example, this is one of the focal planes. It has a local maximum here and here. It has a 0 at the focal plane, and it has shoulders here, and this one that we cannot see. So this 0 means we have 0 information at the focal plane, which translates to that depth discrimination problem. So the reason that we get this nice and flat plan here is because we can actually compensate for the 0 of the film of the first plane with the peak of the film of the second plane. So we somehow maximize the Fisher information matrix, which means that we're minimizing the plan. But obviously the flatness of that curve depends on the plane spacing and the number of planes, as I explained before. So next I'm trying to talk about those two cases. But I would like to stress a little bit more that the design criteria for are actually these listed in here. We would like to have a constant plan along the z-axis because it allows us to localize a single molecule with a constant level of accuracy. And also we would like to have low numerical values because lower the numerical values, higher the accuracy. So let's try to look at the first question. What we're going to do here, we're going to change the plane spacing and look at the results to make a decision which one is better. So the left-hand side panels show the axial plan versus the object Z position, and these are the axial film versus the object Z position. And again, the dashed lines, if you can see them, are the focal planes, the position of the focal planes. So we start with a very small spacing, 0.1 micron. And in this case, as you can see, we have a large peak in the middle of the black curve. So it's not constant, so we get bad, localization accuracy here. And the reason is that if you look at this plot, that red and blue curve, they don't compensate for the zeros of each other properly yet. So we increase the plane spacing, we get better results. And at some point, which we refer to it as the strong plane spacing, we get a very flat curve. Turns out to be 0.5 microns for this set of parameters that we used. Now the reason, so the way that we define this strong plane spacing is based on this curve. So this is the definition. We set the plane spacing to be such that the local maximum of the film of one of the planes overlaps with the 0 of the film of the other plane. So in this way, we maximize the Fisher information matrix. So now if we increase well, probably this is the best spacing that we can get, but if we increase the plane spacing, we start seeing those peaks at the focal planes, and we also name. We also set the maximum plane spacing to be the case where so we have that shoulder, right? Where the shoulder of the film of the second plane overlaps with the 0 of the first plane. And why we're making this definition becomes more clear if you look at the other one. Because from this point onwards, if we increase the plane spacing further, we end up with very bad results. For example, for 3 microns, we have very large peaks here and here. So the depth discrimination problem is back, and the reason is that if you look at this curve, the film of the second plane, the contribution of that is minimal on the 0 of the film. of the first plane. So, so overall, a small plane spacing and a large plane spacing are not desired because we don't get localization accuracy. And the good ones are the strong coupling spacing and the weak coupling spacing scenarios. So these are all based on the axial plan, but we are also interested in making sure that the behavior of the lateral plan is also fine. So this is what I'm going to talk about next in this slide. But just to be clear, this lateral plan characterizes the lateral localization accuracy of the single molecule, which is associated with the detectability of the particle. So if you have a poor lateral localization accuracy, it means that it's very difficult to visually detect your particle in your image. And so they are very related to each other. So we again start with the small spacing. The left-hand side panels are the axial plan, just copied from the previous slide for reference. The middle ones are the lateral plan, which we're studying now. And these ones are going to be the simulated image of the single molecule at this position, which is actually in the middle of the distance between the two focal planes. Again, the dashed lines are the focal planes. So with very small spacing, as you can see, the numerical value of the lateral plan is small in here. And we get a large peak for the particle, so the particle is detectable. We increase the spacing to 0.5 micron. Still, we have low numerical values in here, and the particle is detectable. When we increase the spacing, at some point we start seeing a small peak in here. At that position, with that spacing, the particle becomes marginally detectable, and the noise becomes more significant, so we can barely see the particle. And it turns out this spacing is the same as the weak coupling spacing. So if we even further increase the spacing, as you can see, the numerical values of the lateral plan become so large we cannot detect the particle anymore. So actually this is very interesting and helpful because this weak coupling spacing gives us a guideline as to how to set the maximum plane spacing in our experiment. We know if we go further, beyond that point, we cannot detect our particle anymore. So that's a limiting factor or a guideline for setting the maximum plane spacing. So I also would like to mention these are all explained for a two-plane mount setup because it was easier to explain, but we could easily extend them to multiple planes, which we have done, but I'm not going to talk about it here. The next question that we wanted to address was the appropriate number of focal planes. So this is very brief. So we are trying to find the appropriate number of focal planes to cover a sample of a certain thickness. Let's say this is our sample. So we already know the best spacing that we can get is the strong coupling spacing. So we pick that spacing and keep it constant. So the spacing is going to be always the same. We change the number of planes now. So for example, we could have 2 focal planes, we could have 3 planes, still the spacing is the same. Or we could have all the way up to 8 focal planes, for example, or more. The other important thing is that we assume that the total photon count is fixed, which is the case in practice. So we have a fixed and limited number of photons in practice, independent of the number of focal planes. And we just split it among different focal planes. So this is a practical assumption. And here are the results. This is the axial plan versus the objective position and the lateral. As you can see, with a small number of planes, for example, 2, we get large variations in the curve, or also here. If we increase the plane spacing to 8, for example, we get a very flat curve. So depending, and also notice we get the flat curve, but the numerical values of the plan is large, so the accuracy on average is not good. So what is the appropriate number of planes? It actually really depends on the application. If you are interested in a very flat localization accuracy, we pick a large number of planes, but in order to compensate for that, I guess, large numerical values here, we need to, for example, collect more photons. And for other, for example, experiments that we need to have very good accuracy for a specific region in the sample, we go with a smaller number of focal planes. So with this, I would like also to mention our software package. As we've seen, calculation of this film and the plan is usually difficult and also computationally expensive. And also it depends on many parameters that I did not mention here. For example, the numerical aperture of the objective lens, magnification and everything. So we came up with a software package called the MOM Design Tool. This actually considers all the practical parameters and everything that we need, and it helps you to design the plane spacing. And it has 2 working modes. The rapid mode is based on an approximation, which we mentioned in our paper. It allows you to set the plane spacing in real time and visualize your plan and the Fisher information. And also, then the precise mode models, including the background noise, without noise, and electron. And it's available online on our website. So I'm going to skip the summary, and I would like to thank my advisor, Professor Robert, who's here, Professor Ward, and Shukov, Jerry, and Anish, and Felix, my colleagues, which helped me a lot with this project. We are supported by NIH, and all the details can be found in our paper listed in here. And that's thanks for your attention.
One short question.
So spherical aberration also produces an asymmetry. Have you looked into how that impacts this?
So currently we use the Bohr-Envelope PSF model, but our framework is very general, so we're trying to use other more practical PSF models, and we can incorporate it in the software package.